Everyone, and thank you so much for joining us today for the first session in the Royal Irish Academy series on COVID-19, Perspectives and Pathways in a Time of Crisis. My name is Daniel Carey, and I'm Vice President of the Academy and Director of the Moore Institute at NUI Galway. The inspiration in devising this series has been to further the RIA's mission as an institution committed to discussion and debate in order to advance the public good. The need for shared knowledge and understanding has never been greater as we contend with the crisis and plan for the future. Next Wednesday, we will be discussing in the second of our series, the economic crises past and present, lessons for Irish policy, and then on Wednesday the 15th, COVID-19, social justice and rebuilding Ireland. You can find out more information on this on the RIA's website. Over 330 people have signed up for today's event. I'm very grateful to Catherine Gonson for chairing and convening the session with the leading experts in the field to discuss, we're all virologists now, the science of COVID-19. Catherine is Science Secretary of the RIA and Professor in the School of Medicine at UCD. A quick word of thanks to Jennifer Keneally at the RIA, who is running the show behind the scenes today. So Catherine, over to you.
Okay, great. Thanks, Dan. So as chair of this afternoon's session, I'd also like to join Dan in welcoming you virtually to Academy House for the inaugural conversation into perspectives and pathways in a time of crisis. So the focus of our conversation today will be the science of COVID-19, and we're very fortunate to be joined by a panel of international leaders in the field. So we're going to deal with the origin and nature of SARS-CoV-2 virus, vaccine development, the impact of tracing and tracking infection, and the evolution of our knowledge and its impact on disease and resource management now and into the future. So our panelists are going to speak for 5 minutes around key developments, and after they've finished, we're going to have a discussion. So questions from the audience are very welcome, and ideally we would like these to be submitted through the chat function on Zoom. So we'll have a discussion once all of our 5 panelists have concluded their short presentations. So I'm delighted to welcome our first panelist, who is Ultan Power. Ultan is Professor of Molecular Virology at Queen's University Belfast. So over to you, Ultan.
Sorry, I seem to have difficulty with sharing my screen. I think the host has disabled that. Was it possible to sort that?
It should be okay now, Ultan, I think.
Okay. No, it's still telling me host disabled participating screen sharing.
I might share it for you. Is that okay?
If you could, that'd be good. Okay, good stuff. So if you have the first slide, then please, Jennifer, that'd be great.
Yep.
Okay, so just to get started then very quickly, just to give you a very brief overview of the timeline, the very early timeline of SARS-CoV-2 and its discovery. The likelihood is that the first case happened in mid-November in 2019, as the first symptomatic case was traced back to December 1st, 2019. By December 8th, there was a series of patients with pneumonia of unknown etiology discovered in Wuhan in China. And by the 31st of December, the WHO were notified of an outbreak in the Wuhan region. And it subsequently developed from there as an epidemic, first of all, an outbreak, first of all, then an epidemic, and now subsequently a pandemic. The major achievement, I guess, with the early stages of this virus was marked on January 7th, when the new coronavirus was identified and the full genomic sequence was published and shared with worldwide. And this really set in motion then a lot of the work that was done subsequently in terms of trying to identify the origin of the virus, but also in terms of vaccine development and trying to identify the pathogenesis of the virus. Now, today, as of today, there's over 10 million cases that are currently active and over 500,000 deaths. So the consequences of it has been major over that period of time. Do you have the next slide, please, Jennifer? So just to give you a brief overview then of the virus itself. So it's a positive single-stranded RNA genome. It's just under 30,000 nucleotides, the whole of the genome. It's an envelope virus. The size is ranges quite a lot, from 50 nanometers to 200 nanometers in terms of diameter. And it's composed, the actual structure, the coronavirus structure itself is composed of 4 structural proteins, but the genome encodes for 25 other non-structural proteins, which have a whole bunch of functions that I won't go into here. Next slide. So the classification of the virus then, it's a part of the Nidovirales, Coronaviridae family, subfamily Coronavirinae. It's a beta coronavirus, like some of the circulating human coronaviruses that are endemic every single year. And it's also a member of the Sarba-virus. It's related to other viruses like the bat coronavirus, and I'm going to show you some phylogenetic trees associated with these, or breakdown of these in terms of relationships with particularly the bat coronavirus and the pangolin coronavirus as well, okay? But other viruses that are present in this family include the SARS-CoV from 2003 outbreak and the MERS-CoV, which was first discovered in 2012. The other common human endemic viruses that are known are the OC43, HKU1, 229E, and NL63. And these provide a lot of information about how coronaviruses in general interact with the human in terms of repeated infections, in terms of duration of immunity, etc. So they provide some evidence or some indications for us of what we might expect in terms of, say, the duration of immune responses to SARS-CoV-2. Next slide, please. So this gives you then an indication of the association, or the cousins, as it were, of the SARS-CoV-2. So in the yellow, you have the Sabra virus, co-viruses, indicating a lot of the SARS-CoV-2 strains that have currently been sequenced. There are many, many more available at this stage. But what you'll see from there, that the most closely related virus is our bat coronaviruses, okay? And in particularly the, sorry, next slide from that, please, is the bat or G13 strain, okay? So the overall sequence homology between the bat coronavirus, this particular strain, and SARS-CoV-2 is approximately 96%. So it gives a very clear indication that these viruses are very, very closely related. Now, what's very interesting, though, is if we look specifically at the spike protein associated with this, spike protein gene associated with this virus, what's really interesting is that the bat or rat G15 or G13 actually has quite a lot of differences in that region. But if we compare it with the pangolin from Guangdong region in China, we see that it's completely identical. There are no changes at all. And this then is where the first suggestions were made that maybe this virus has passed through or somehow interacted with pangolin coronaviruses. And again, similarly, if you look at the receptor binding domain, which is part of the spike protein, again, you find that there's a complete homology, except for one amino acid change, there's a complete homology in the receptor binding domain between the Guangdong pangolin coronavirus and the SARS-CoV-2. However, the level of identity between, at the overall genome level for the Guangdong pangolin coronavirus is about 83%. So it suggests that this might be indicative of some kind of recombination event that's happening, perhaps in the pangolin as an intermediate host between the bat and humans. Go to the next slide, please. So this is what I think people are currently thinking about in terms of the origin of the SARS-CoV-2. One hypothesis, and I should stress that we actually don't know for sure what the origin of the SARS-CoV-2 is. There's still a lot of information, a lot of research that needs to be done to try and trace exactly where it came from. But the current thinking is that the bat coronavirus, RATG13, is found in horseshoe bats. It's a very, very close homology to the SARS-CoV-2 virus, except for this receptor binding domain in the spike protein. It suggested that the bat coronavirus then passaged as an intermediate host into the pangolin, where you may have had a recombination event that recombined the pangolin coronavirus receptor binding domain from it into the bat coronavirus, and from there that may have infected then humans in, for example, the wet market in Wuhan, China, for example. That'd be one possibility of having exposure to these animals. The alternative is then that the bat virus infected the pangolin virus, the recombination event happened, and then it reinfected the bat, and that from there may have gone into the human species. Or a recombination event may have happened alternatively where the bat went directly into a human virus. Now, one of the big issues in terms of trying to determine or get further information about the origin of the virus is that the bat coronavirus was isolated in the Yunnan province in China, which is over 1,000 kilometers from the Hubei province, where the SARS coronavirus was first isolated. And similarly, the pangolin coronavirus from Guangdong is 828 kilometers from the Hubei province. And this then gives geographically some difficulty in trying to comprehend where the origin of actually these viruses will be. Next slide, please. So I just wanted to leave you this last one then in terms of the current evolution of the virus. Overall, the virus seems to be evolving extremely slowly. There is one interesting divide, though, that seems to be happening between the viruses from different regions, and that is this G614D mutation in the spike protein, which seems to differentiate between the Chinese and European strains that are currently circulating. And one question mark related to this virus then, is it possible that this G614D mutation is associated then with increased infectivity in Europe compared to what actually was happening in China? Overall, as I say, the SARS-CoV-2 evolution appears to be slow. Most of the mutations that are happening appear to be non-synonymous, so they're not having an impact on at the amino acid level, but they do allow the capacity to be able to trace the virus from different origins, for example, from China to Iran, Iran to Italy, and Italy then into Europe. There are many other countries in Europe. But overall, from an evolution point of view, it seems to be very low, which I suppose is good news from the point of view of developing a vaccine eventually against this virus. So I'll leave it at that, Catherine, and I'll hand it over to you and the next speaker.
Okay, great. Thanks a million, Sam. That was super. So, sorry, Ultan. So our next speaker is Professor Sam McConkey from the Royal College of Surgeons, and Sam is no stranger to anybody watching RTE these days or any other media. So Sam is going to talk to us about tracing and tracking and its importance in understanding and hopefully controlling COVID-19.
So thank you very much, Catherine, and thanks for the invitation to speak to you and to everyone. I don't have slides. I'm going to talk quite briefly. I'd like to focus really on what I see is really a very contested issue now, which we could call epistemology or theory of knowledge. And it's how do we know what's going on? Because it's quite clear in all of this that many people are contesting the role of experts and contesting the role of science and using alternative sources of information. And that, in my view, is one of the biggest challenges that those of us trying to control this face at this time. I'm going to maybe outline some of my own background and journey as a sort of chronological narrative for how this has evolved, and I hope that highlights several of the issues around testing and maths and knowledge. So I'm a medical doctor and specialist in infectious disease. In fact, last night the Infectious Disease Society of Ireland has now elected me their president, so I'm very pleased with that. What's a bit unique is that while doing a fellowship in St. Louis, Missouri and WashU, I did a master's in mathematics and wrote a couple of grants to try and get funding for mathematical modelling. Now, neither of those got funded. I ended up doing vaccine development instead of that, but I had large background in maths, in spatial epidemiology, in statistical inference of spatial data, and applied to the US and to Egypt. My background is also a bit in business. I spent 5 years on the board of HICWA, which was a wonderful experience of learning how the Irish cell system both works and doesn't work. I've also done a lot of private sector consulting and also for peer review of research funding proposals in tropical medicine for groups all over the world, Danish, British, French, European, EU, Irish, American. So a lot of critique of other people's proposals for tropical medicine research suggestions, which has been very helpful in trying to understand the limitations of research and science in a practical way. So over the years, many of us in ID use a information source called ProMed, P-R-O-M-E-D, from the International Infectious Disease Society. And that sends me an email every day and tells you about 5 or 6 new outbreaks that happened in the last 24 hours. And of course, in the end of December, we got a little flag that there were some pneumonia in Wuhan, which, you know, doesn't really raise much of an eyebrow because you see these regularly. I've obviously been watching influenza and watching Nipah virus and many other viruses, sort of in the expectation that every year or two, one of them is going to sort of develop human-to-human transmission and go rampant and perhaps have a high mortality or perhaps have a low mortality. So my attitude to it at the end of December was just sort of watch and see. On the 12th of January, then the Chinese told us that this was a coronavirus, which of course, given MERS and given SARS, MERS had a 40% mortality, mostly in healthcare workers. Anyone who's an infectious disease physician sort of sits up and says, oh, this could be a coronavirus, right? That's 40% mortality and a lot of transmission in a healthcare setting because our own existence is threatened by those sort of rather horrible things. And SARS obviously came to Ireland in a single case, but didn't spread here due to the good work of the people in Ballinasloe, but it did spread a lot in Toronto and Hong Kong. And of course, Margaret Chan largely dealt with it very well in Hong Kong before she became head of WHO. So I was very aware that a coronavirus is potentially a very, very bad thing if it can spread to humans effectively, and if it has a high mortality or even a 1% mortality is still a problem. I then believe in watching what people say about themselves. So started looking at the Chinese websites in Chinese language of the Wuhan CDC, of the Hubei CDC, and of the China National CDC, using Google Translate to translate those into English, and then looking at the quantitative data of number of cases for both Wuhan, Hubei, other cities in Hubei, and for other provinces in China, and started to model that in spreadsheets with mathematical models. So there are a lot of complex models you can use for infection, but when you don't know what you're dealing with, my advice is to use really simple models. The beginning of an epidemic typically grows with a doubling time in an exponential curve. So I took the raw data, worked out number of new cases per day, logged the data, simple log transformation, and then used linear models of the log data to try and look at the slope, look at the growth rate. And that because we knew that the intergenerational time is about 5 days, you can work out an R value, a transmission rate from one case to how many secondary cases will occur using that model data. And it was clear initially the R value was somewhere between 2 and 3 in China. What was really shocking was the death rate in Wuhan was 4 or 5%. So with caution, I watched this spread to other Chinese provinces and then shockingly spread to Singapore, South Korea, Japan. And at that stage, in sort of second, third week of January, had the sort of dawning realization that this could be a massive worldwide pandemic. That was confirmed on the 23rd of January when the Chinese government closed all non-essential industry in Wuhan. They switched off about 80% of their economy. The next day, on the 24th of January, the Chinese Communist Party, Xi Jinping, closed the rest of the province of Hubei. That's 55 million people. All the economy switched off except for essential. Closed off roads, trains, airports. And on the next day, the 25th of January, they closed off non-essential industry and travel and activity in all of China. So, you know, the sort of second biggest economy in the world essentially closed down. What that told me was that the Chinese Communist Party had seen with their own eyes immediate and present danger of an existential sort to the Chinese people and to the Chinese state, to the extent that they were willing to switch off essentially 80% of their economy overnight. Because of SARS-1 and because of MERS, almost all Asian countries had a plan. They had a pandemic coronavirus plan on the shelf. And it was clear at that time that the Chinese had jumped from doing nothing or very little to the highest level of their implementation of a coronavirus control plan over a couple of days. So it went straight to the highest of the high levels in the coronavirus. There's almost nothing you could imagine that could have been done in China to control this virus that was not done. They ramped up contact tracing, essentially had house arrest for the population. They had everyone wearing masks, and they ramped up testing for the virus in a very, very widespread scale. So the whole of the Chinese economy, 1.5 billion people, was transformed over 2 days to deal with one item, which was this coronavirus that was spreading in an uncontrollable fashion in Wuhan and killing 4 or 5% of people there. Once it started spreading to other countries, and once it was clear that it wasn't being controlled in China, that it had escaped and was going to spread widely, then of course it was inevitably going to come to Europe. So again, using ProMed data, I started to model basically all the countries in the world. Obviously the Diamond Princess was an interesting experiment. It showed us that there is about a 1% mortality, and that the attack rate, you know, was around 20% of the people on the ship got it, and it didn't stop transmitting until they took them all off the ship. So obviously, I love sailing, as you all heard on Monday night. Sailing on a cruise ship is not a good way if there's one person on the ship with the virus. So my simple, robust models then moved on to look at a website called Euromomo, EUROMOMO, set up about 5 years ago by the European Union. It models weekly absolute all-cause mortality in about 25 European countries, split up by age group. And it was quite clear then, of course, in Northern Italy and Spain, that there was a striking rise in mortality for all causes in Italy and Spain. That subsequently spread to UK. And UK, ultimately, the all-cause mortality in the UK hit about 45 standard deviations above the expected one. Most of the listeners will be familiar with 6 Sigma quality control methods from Japan. Once you're 6 standard deviations away from the expected mean of a process, you're out of control and you need to fix it. So Britain's mortality was 43 or 44 standard deviations away. The Italian was about 20, and ours was about 4 or 5 in Ireland. So this was not just impacting the known cases that were diagnosed with PCR. What was clear is you have to look at all-cause mortality because there are obviously lots of people who die without diagnosis. I then started looking at segmented data by state, by province in Italy, because there's great heterogeneity by location. We've seen how New York was hit early, whereas now California and Florida have been hit later. So to try and divide both Ireland into county level and US by state and Italy by province, to try and look at how the disease is spreading in different regions. So that's really, I continue to do that. There's good data from the HPSC. It's helpful in terms of analysing data to know the source of the data really well. it's necessary to know the limitations. For example, every time our testing criteria in Ireland changes, we can't compare the rates after to the rates before directly, because the testing criteria will obviously change the numbers. Similarly, as we scaled up diagnostic testing, it was clear that obviously the number of cases will increase. I know President Trump then seemed to say that if we tested less, we'd have less numbers, which is one interesting way. I think that's called the ostrich approach. Let's bury our head in the sand. So I'm not recommending that. But when you're doing mathematical modeling, the rate of availability of testing clearly changes the numbers. And the way to deal with that, of course, is to look at the proportion of tests that are positive. So you can look at the proportion positive, and that data, is available from the HPSC. So that's really my view of the models and the numbers. I'm hoping that really social control of this virus and preventing its spread through changing our social interactions can control it. I'm very pessimistic. I worked on malaria vaccines, hepatitis B vaccines, a lot of vaccines for many years, and the product cycle time for developing vaccines is about 7 years, in the human cycle about 3 years, in the animal cycle where they cut all the corners. So this idea of a vaccine in 6 months, I'm good friends with Adrian in Oxford who's saying this, but it's the sort of best to best case scenarios. It's every experiment works well the first time, and those of us who've done a lot of clinical trials know that they don't always work the first time. You need some dose ranging and some looking at adjuvants, looking at routes, looking at optimizing the protein and the antigen and so on. So I see a vaccine coming, hopefully, maybe in a number of years' time. I'm positive about remdesivir. It's a little signal, but at a test tube level, it's effective at about 20,000 higher concentration than the drugs we use for HIV or Hep C. So our Hep C drugs are now good at 1 nanomolar, whereas this is about 20 micromolar effective concentration of remdesivir to inhibit the replication of coronavirus. Obviously, dexamethasone works. I haven't seen the paper yet, but I believe it. It's partially effective, takes down the immune response. But no amount of intensive care, no amount of good drugs, no amount of steroids and antivirals will control this, because many people won't be able to access them quick enough. So to control it in the 8 billion people out of the world, we need the traditional methods of epidemiology and public health infectious disease control, like cordon sanitaire, physical distancing, like masks, like reorganizing how we travel and how we live our life. So I've tried to do some sort of public health motivation of our people in Ireland. This has to be done at nation state level. At no other level have we got the influence to control people. If someone in Brussels was to say do all this, no one would listen to them. Or if the UN or WHO were to say it, no one would listen. So it has to be led at national level. It does require national unity. I'm delighted your RIA is doing a thing on social cohesion. Without social cohesion, if only half the population follow these measures, it isn't going to work at all. So you've heard me, I don't know, Sam the Socialist. That was read out in the Dáil as Sam the Socialist, which I'm not upset about that, but I'm probably more in the green variety than the pink one. But anyway, for this virus, we certainly need social cohesion and buy-in from everyone. If that means a better deal for housing for our teenagers and young people and more access to education and healthcare, then that to me is also a health good. Social inequity is bad for health. We know a lot about Michael Marmot's social determinants of health, and it's not just about the wealth of a society that determines its health, it's the spread of that. So countries like South Africa that are very inequitable tend to have poor health outcomes when you look at their GDP per capita. Similarly, countries like Cuba that are very equitable have better health outcomes, even though they're not particularly rich. So there's a very strong second principal component, if you like, to the outcomes health that equity leads to better outcomes. So I'm comfortable with that message as a doctor and as a public health advocate. So thank you. I'm happy to take questions.
Thanks very much, Sam. Can I just remind people, if they want to submit any questions to the panel for our discussion after later on, they can do so through the chat function on Zoom. So our next speaker is a colleague of mine from UCD. So Paddy Mallon is Professor of Infectious Diseases Medicine and a consultant in infectious diseases at St. Vincent's University Hospital. And Paddy's going to talk to us about how clinical management and therapeutic interventions are evolving in treatment of COVID-19. So we've heard mention from Sam about steroids and other sort of anti-inflammatory strategies. So we look forward to hearing from Paddy now. Thanks, Paddy.
Thanks, Catherine. Can you see these slides okay? presume so.
Yep.
Yep. So just to go through, just to follow up in terms of what Sam said, as with all new infections, pretty much whatever you thought you knew about a coronavirus, you can more or less throw in the bin. And the same goes for SARS-CoV-2. The conventional approach to management of any infection is that you have the pathogen, you have the host develops the disease. Broadly speaking, two outcomes, either a favorable outcome or a non-favorable outcome. And the goal of treatment is to try and steer the risk more towards a more favorable outcome. And that's your sort of conventional wisdom. And that's been a lot of the approach of the big trials, the big platform trials that have been introduced for SARS-CoV-2, like Discovery and Solidarity and Recovery. But it's not that simple. COVID-19 infection is a lot more complex, and there's a lot of different stages of the infection. You have the initial infection itself, and one of the big aspects to that is the ability of this virus to transmit to other people. And as Sam said, that's going to be an important component in terms of therapies to try and prevent that transmission. But not everyone who gets the virus gets sick. Some people get very little of no symptoms. Some people end up in hospital. And even those that are in hospital, a smaller proportion will end up developing quite severe infection that will require either ICU admission or non-invasive ventilation. And at each of these stages, people have the ability either to recover spontaneously or, unfortunately, significant proportion do end up succumbing to the infection. So in terms of managing this type of approach, what we need to be able to do is to develop treatments that will be best targeted to a specific pathway in this infection. And the reason that's important is that we not only have the infective stage, but we're also recognizing this concept of post-COVID recovery, where the recovery from this infection is not the sort of usual recovery that we see with other simple coronaviruses. And again, probably underlies unique characteristics of the interaction between this infection and the host. So the treatments, to understand where the treatments are best placed, I think we've got to have a greater understanding of the underlying pathogenesis of what's driving each step within this disease process. And what's starting to emerge now is that the actual role of the virus is probably more focused in this early phase, definitely driving transmissions and probably driving some of the early symptoms. But what we're also recognizing and accumulating amount of data is being published on this is the importance of the host immune response, specifically in terms of driving this more severe phenotype that leads people to end up in hospital and into ICU. And it's probably also driving some of the post-COVID symptoms that we see. Another aspect that I find is very unusual, I think almost unique to these types of viral infections, is the role of coagulation disturbance within SARS-CoV-2 infection. And we're seeing a lot of probably the disease and outcomes that are being driven not by the virus and perhaps not directly by the host immune response, but because of coagulation disturbances. So large vessel thromboses, end organ damage, and a number of different organs. And as anyone gets unwell, you do have end organ damage, and that can be contributing both to overall mortality and morbidity together with this slow post-COVID recovery. So trying to figure out how best to treat a condition like this, we've got to understand which treatments best fit into which section of the disease process. And there are a huge number of potential therapies currently being studied. So over 2,300 individual studies are registered on clinicaltrials.gov, and this gives you some idea of the types of treatments that are being investigated. So antivirals, immunotherapy, so this would be modifying the host immune response, anticoagulants, looking at that coagulation difficulty, and then some additional therapies to try and prevent this end organ damage. And what we needed to do is to be able to drive research in all of these areas. When you look at how many of these drugs so far have been shown to work in randomized controlled trials, we've got 2 altogether. And dexamethasone as an immunotherapy has actually been shown to work best within the ICU setting. Remdesivir, some early data to suggest that it might modify overall length of stay in hospital, but no mortality benefit. But we're also starting to see a lot of drugs fall off this list, and I think as time goes on, we'll probably see more and more drop off the list. So we have a really limited data in terms of what we should be using to treat this infection. So how do we go about getting the data that we need? Well, this is the pathway I would usually use to look for clinical discovery with any new condition that's coming along, and I think SARS-CoV-2 and COVID-19 infection are no different. We're really at the very early stages of understanding this disease, and it's only really through understanding this disease that you can model associations with the disease and identify issues such as hyperimmune reactions, abnormal coagulation, and then you really need to bring that into the lab, try and tease out these mechanisms in a lot more detail and find pathways that are druggable. And this should really be what drives our therapeutic targets going forward. And at the moment, I mean, my experience, this process usually takes anywhere up to 5 to 10 years from start to finish, but very rapidly, we're quite a way down this pathway, and we're starting to get some really nice translational information through on this infection that I think will help speed up more directed therapies. How are we doing this in Ireland? Well, one of the important components of driving any early translational research model is having cohorted, well-defined and well-phenotyped cohorts that you can do these association studies that then drive a translational program. And we're doing this within Ireland through the All Ireland Infectious Diseases cohort. So this is something that has developed from a cohort that we've been running in UCD now for upwards of 10 years, directed against HIV and other infections. And we've now been able to rapidly expand it to hospitals around the country. So we now have 11 different centres around the country, including most adult hospitals and paediatric hospitals. It's coordinated through the UCD CRC, and Peter Doran has been central in terms of linking in with the resources of the CRCs around the country to drive this programme forward. Fairly simple concept. You have a study that's been approved by NREC, so we've been able to rely on some of the good things that have come out of the COVID pandemic so far. Really, it's an open eligibility for recruitment. Any infectious disease related activity, people can be recruited. They can consent for routine data for research. We have an online CRF to collect our phenotyping data, and then an extensive biobank that enables us to really look at a number of different translational targets and pathways, and with robust data access guidelines. And very quickly, we've been able to leverage a lot of funding through our basic science colleagues. It's really driving a lot of the core research in the field. It gives you some examples here of some of the funding calls, or some of the funding awards that have been made by SFI and HRB that have been linking in with the All Ireland Infectious Diseases cohort. And you can see upwards of €1.6 million in predominantly basic science research, looking at anywhere from viral sequencing through to microbiome, through to coagulation studies and potentially new antiviral compounds, all being driven by this core of a well-phenotyped population linked to a detailed biobank. And we've managed to do all of this without any core funding. This has been really leveraged on a pre-existing infrastructure that was built up by the HRB over many years. And I'm hoping that going forward, that the powers that be can find a way to provide core funding to keep this type of project going, because it's going to be the fundamental core of our response to pandemics leading into the future. That's all I've got to say. I'd like to thank you for your attention, and I'd look forward to some questions later in the session. Thanks.
Thanks a lot, Paddy. That was really great. So our next speaker is Kingston Mills from Trinity. Kingston is Professor of Experimental Immunology, and he's going to talk to us about vaccines against SARS-CoV-2.
Okay, good afternoon, everybody. Hopefully you can see my slides. Vaccines are obviously the solution to this problem, as Sam said earlier in his talk. While we might be a long way to quelling the first wave of this infection, if we do get a second or subsequent wave, we definitely need a vaccine, and it's by far the best way of controlling this. The good news is there are about 150 vaccines in development at various stages. 17 of these are already in clinical evaluation, some in as far as phase 2, and one moving into phase 3 pretty soon. It's the quickest development of a vaccine that's ever been seen, and I'm going to come back to this in a minute. But first of all, just to tell you the type of vaccines that are out there. Traditionally, vaccines, there are 7 different approaches for making vaccines. the early days, it was all inactivated whole virus or live attenuated vaccines. And more recently, recombinant or subunit vaccines have, where there's an antigen from a virus, for example, influenza hemagglutinin, we would example here, synthetic peptides, still experimental. And then 3 other ways that are really still unproven, and these are 2 of these are being used as a means of generating candidate vaccine against COVID-19. One is using the nucleic acid from, or the messenger RNA from the virus, and actually injecting the messenger RNA or the DNA, and the other is putting the messenger RNA into a vector. And the vector that's most commonly used is the common cold adenovirus, because it's a safe virus and it induces a good immune response. So in terms of what COVID-19 vaccines are currently in clinical trials, this is a selection of them. Probably the first ones to get going were the Chinese ones, and these were based on the old-fashioned inactivated whole virus technology, and these are looking pretty good. Some of the animal data, in particular the animal model data, is looking very exciting from the whole virus vaccines. The problem with this is that production of the vaccine is slow and it's laborious, and you can't generate billions of doses of it overnight, but the good thing about it is it's a proven technology shown to work. Then you have the adenovirus, and there's a number of groups here, including the CanSin group in China, but also the Oxford vaccine group in collaboration now with AstraZeneca, and they've have very bullish timelines of 6 months, of which most of us don't believe is going to be achieved. But you know, fair dues to them for being enthusiastic about it. And then probably the biggest focus has been the messenger RNA vaccines. Moderna were the first company to get a vaccine into clinical trials. It took them 42 days from the sequence of the virus to getting a vaccine ready to go into clinical trials. That's unprecedented, absolutely incredible. And the NIH are funding this work, and they're now in phase 2, and they plan to be in phase 3 before the end of the year. Imperial College groups in Germany are also working on the same approach, and then DNA approaches to 2 companies in Korea. And finally, recombinant with an adjuvant. There's a couple of companies, one in Australia, one in China, in collaboration with GSK and Dynavax. These are 2 traditional vaccine companies. And this is probably a safer approach. If I had to put my money on which one I thought was going to work, I would say this one here, the inactivated virus. These are unproven technologies that may or may not work. If they do work, the big advantages are you can produce very large amounts of it very quickly. So you could, in theory, make hundreds of millions or billions even of doses in a few months, and that would be one significant advantage. So in terms of timescale, this is the timescale of a typical vaccine, 10+ years. You target, you develop your antigen, you know what your protective mechanism, you go into phase 1 testing for safety in a small number, phase 2 testing for safety in a larger number, phase 3 efficacy testing, does it actually work in the field, and then scale up manufacturing and licensure. Normally it takes 10 years or more even. This is what they're hoping to do. This is a slide that I've taken from a seminar that Tony Fauci gave, and where it shows they're hoping to condense this into about 18 months. So it is particularly incredible by combining phase 1 and overlapping it with phase 2, and combining and overlapping phase 2 with phase 3, and starting the manufacturing process before they know that the vaccine is going to work. And a number of companies, including AstraZeneca, are doing this already. They're building plants to manufacture the vaccine without knowing that this vaccine is actually going to work. That would be unheard of in the past. So there is some realism about the timelines in terms of maybe 18 months, but I think 6 months is over-optimistic, and certainly not going to be 10 years. If any of these works, of course, that's the other issue. So how does a vaccine work, and what are they trying to do? The key thing is generating what we call virus neutralizing antibodies. When you inject a vaccine, the innate immune cells pick up the antigen, take it to a lymph node, prime T and B cells. You get B cells which make antibody. Those antibodies then bind to, the usual antigen is the SARS spike protein. So they bind to the spike protein. The spike protein normally binds to its receptor, ACE2. If you block that binding, you stop infectivity. And that's the most effective way of getting a vaccine. And that's nearly all viral vaccines in use today work not nearly all work by making neutralizing antibodies. They also require T cells to help this antibody production, but also T cells can themselves function in protective immunity by killing virally infected cells. So there are some vaccines for example, against TB and HIV that have been developed on the back of, there's no HIV one that works, but the candidate ones have been built on the back of making T cell responses. Now, the other thing is, do these immune responses persist? And one of the issues is that antibodies can decline, and it's known that antibodies against coronaviruses can decline. And this is what happens. You get a boost when you immunize a couple of times, you get antibody responses, which with something like measles can last for decades, but other infections, they drop off very quickly. But you do have memory T and B cells which remain for decades. So even if the antibody disappears, those memory T and B cells can be reactivated and then potentially start making antibodies again once the person gets challenged with the virus. So even if the antibody drops off, if you have the memory T and B cells, you may get persistent immunity. So it's something we don't know about COVID-19 yet, and only time will tell if antibodies even protect. I just want to finish by telling you about a project that we're doing in Trinity, which is around the immunology of COVID-19. And this has a vaccine element to it, but it's got other elements as well. I'm co-leading it with Aideen Long from TTMI and Trinity Biomedical Science Institute. It's where I am. I'm the director there. There's a number of leading PIs that are co-PIs on this, and we've got significant collaboration internationally as well as nationally. We've already raised 2.4 million through a very generous donation from Allied Irish Banks, and SFI are hoping to make an announcement tomorrow on this project. So it's all about, more rapid testing methods, looking at the immune responses in patients, both at the inflammation level, but also in patients that have been exposed and didn't become infected, because these may be the clue to finding the way to generate a vaccine. If you can tell those that resisted infections, figure out how the immune response is, and then you can help define the vaccines. We're also working with some of the pharma industry on the vaccine side by helping them, in particular in the area of memory, how we can get a more persistent immune response by vaccinations. That's a little taste of what we're doing in Trinity. And I'll leave it at that and pass it on to the next speaker.
Okay, great, Kingston. Thanks very much. That was great. Our final panelist joins us from New York. So Barbara Murphy is Chair of Medicine and Head of the Department of Medicine at the Icahn School of Medicine at Mount Sinai. Barbara is an internationally renowned nephrologist, and she's going to tell us something about the lessons that we are learning, certainly learning at the moment, and the potential future impact of COVID-19. Thanks, Barbara.
Taking down the virtual background from an event that we had last night. So thanks very much, Catherine, and thanks to the other speakers. Wonderful, wonderful talks by everybody. I am talking to you actually from Vermont as the chalet background.
Okay.
We escaped New York after 3 months, having gone a little stir crazy. We felt we needed to get out, and having got tested negative, left New York and headed to Vermont, where you have to be negative to actually go to the supermarket. So I just want to give you, let me share screen. Share screen. Share. Make this. Okay.
We have you.
Good, excellent. I just want to talk to you about just the lessons learned and long-term consequences, just from the perspective of the Mount Sinai healthcare system and what we went through. So just to give you an idea what I mean by the Mount Sinai healthcare system. So we have 8 hospitals. I'm responsible for the medicine clinical program in all of those 8 hospitals. These are the 4 within Manhattan. The upper left-hand panel is Mount Sinai on the Upper East Side, which is where the big brown, ugly brown building, as we call it, is mainly a research building, as is this other building over here. And this is all clinical care here. Oops. The upper right-hand panel is Mount Sinai Beth Israel, which is down at 1st and 17th Street. Bottom left is Mount Sinai West, which is over near the Lincoln Center, and then Mount Sinai St. Luke's, which is up in Morningside, it's up near Columbia University. So there are the 4 main hospitals we have in Manhattan. In addition to that, we also have Mount Sinai, the New York Eye and Ear, which was not really affected by this. It wasn't admitting patients, but it was affected by it in that it had to shut down all elective procedures, and all of their physicians then came to help us. In addition, we have over here number 4, which is Mount Sinai Queens. We also have Mount Sinai Brooklyn, which is off over here somewhere. And on Long Island, we have Mount Sinai South Nassau. So we argue about whether we're one of the biggest or the biggest healthcare system in New York. The other would be Northwell Health, but certainly in Manhattan, we're the biggest healthcare system. So this is what we were looking at the end of February, beginning of March, where Beth Persha in the Department of Medicine was doing modeling to try and predict what our peak would be so that we could try and map out our response. And, you know, I had the end of February, beginning of March, just on simple math, looking at the number of patients we had that were COVID positive and the doubling time, which at that point was 2 days, projected by the 3rd week of March that on the Upper East Side, we would have 750 patients. We, on our inpatient service on the Upper East Side, would normally have 200 patients. So I was trying to, and I did this for each of our hospitals and was mapping out like 200 to 300 patients at each of our other hospitals, and was trying to look at the number of teams we would need on the inpatient service to be able to respond to that. And I will be honest, the docs were like, the head of our hospice program was going, there's no way. And I'm like, yes, we need to plan for this. And if we're wrong, fine. But if we don't plan for it and we're wrong, then it would be really bad. At that point, when I said 750 patients, Beth was actually projecting for our healthcare system that we would have 22,000 patients, which was insane, and just kind of paralyzing to think about. So this is some of the other modeling that she was doing to help us project. So some of the things we started doing, how do we just totally change and reinvent a healthcare system to focus on one disease? Clearly testing was testing. Looking at supplies, PPE. For New York, a big issue was transport, because all of our workers came in via the subway, which if any of you have been to New York, know that it's totally overcrowded during rush hour. And so we had staff, faculty, nursing, everybody that was now afraid to use public transport. Housing became an issue because we had faculty that were afraid to, staff, faculty, everybody, that were afraid to come in and take care of COVID positive patients and then go home and potentially infect their family. So, you know, we had a whole hotel industry in New York that was shut down. And some of the best hotels in New York started offering their hotels to healthcare workers so that they could live there while they were on active duty. And then a big thing was childcare. Same, I know I watched the same discussions in Ireland as you shut down schools and, you know, start social distancing policies. Childcare became a major issue. You know, a lot of the staff, the nursing, the faculty had no one to take care of their children. We have a childcare facility for our staff. We had to close it down. And so, and faculty and staff were afraid to send their children to childcare. So we had to start looking at childcare. How could we take over childcare responsibilities, and there were major legal ramifications to doing that. A huge part around organizing was everybody wanted to help. So how do we clearly delineate roles, responsibilities, and organizational structures? What was a departmental responsibility? What was an institutional responsibility? So I started mobilizing the department, creating organizational structures, and found that we were duplicating efforts that were happening at an institutional level. So we had to reorganize and kind of merge with what was happening at an institutional level. Communication became key because, as you can imagine, people were very concerned, very nervous, very anxious. So we took the policy that over-communicating wasn't a bad thing. I did weekly rounds with the divisions. I did weekly rounds by Zoom with the residents. So the other thing is, not only did we have those hospitals that I outlined, but there's also hospitals that the residents go to that we had no control over. They also do rotations in the Bronx VA. They also do rotations in Elmhurst, and I'm sure many of you saw Elmhurst, which is a city hospital, was highlighted on the news multiple times for significant problems around PPE and ability to respond to the crisis. And we had residents in there, and I was unable to change how things were organized to maximally protect our residents. So we did things like we organized a room on our floor with PPE, and every time residents were going out, a new set of residents were going out to Elmhurst or the VA, we provided them with a to-go bag with PPE, water, snacks, and other things that we thought they might need whilst they were being deployed. But I have to, the weekly communications with the residents were the hardest communications I had all week, where they were out in some of these hospitals with very little supervision, being responsible for incredibly ill patients, running codes by themselves, and trying to mobilize additional supports, deploy faculty and staff out to those hospitals, even though we didn't have jurisdiction over them, but purely to help the residents. The other thing is that I don't know how this would work in Ireland, but although we have 8 hospitals, we're not cross-credentialed in all of those hospitals, even though we're one system, because legally they're separate entities. So immediately we gave emergency privileging to all faculty for all sites. We had to reorganize our ambulatory platform. We created a clear triage mechanism for testing, for screening of patients before they came in. Every person that had an appointment to come in and see a doctor had a screening phone call or telehealth visit. All routine visits were cancelled, all elective surgeries were cancelled, and we created a triage mechanism to minimize the traffic to our EDs because the EDs were emergency rooms were overwhelmed. Sorry, I've become Americanized, shocker, after 23 years. And so we were trying to minimize traffic to the emergency rooms. We were deploying medical teams to the emergency rooms because some patients were being admitted from the ED and staying there for a long time. So they had to be taken care of in the emergency rooms. So we deployed medical teams to the ED to take care of them there. And so, we had to, at all our ambulatory sites, we set up screening in the lobbies. So the patients couldn't get up and into a room if they screened positive. If they screened positive, they had to be navigated to tents. We had tents at all of our sites where people were looked after and either determined that they should go home or sent to the ED. So we totally reinvented how we organized our ambulatory platform. And I'll come back to this in a minute. We totally reorganized how we organized our inpatient service. The institution created a command center to sort of standardize approaches around all of this. Initially, there wasn't departmental representation in that, that's where the duplication started occurring. And so We had department representation in there, and then we had morning AM phone calls where all of the sites were on, and they had to give an update as to their status and their staffing needs. Up until that point, it was 1-800-GO-BARBARA, which wasn't working. I'd get these frantic phone calls in the morning saying, I need 5 teams and and can you send us residents, please? And I'm like, I cannot. And so we created a system, an institutional system to respond and get teams of doctors out to each of the sites. The department created a system that I will come back to maximize utilization of physicians from other departments, and also a second system overseen by our vice chair for quality and clinical operations to rapidly onboard volunteers and locums from outside. And this all meant that HR and legal were on call around the clock to help us get this done. In addition, there was a major reorganization around research. One, ongoing research was stopped unless it was deemed essential. So if you were an IBD patient on an experimental medication, we continued to do that. We did remote testing and remote monitoring using telehealth. And then we, overseen by Judy Aberg, we initiated a new COVID research task force. We mobilized our Department of Medicine Clinical Trials Office and the Cancer Clinical Trials Office, and any other person in a lab or clinical research coordinator that was not working was repurposed to set up the COVID clinical trials at all our sites. Now, mostly in our system, clinical trials only happens on the Upper East Side. We managed to mobilize clinical trials at 7 sites. So Queens, Brooklyn, South Nassau, Morningside, West, who normally never do clinical trials, were involved in all of the COVID clinical trials. As you can imagine, everybody was screaming to be part of the remdesivir trials. We also got up and going, so multiple different trials. And in addition, our basic scientists initiated a biorepository and a registry. 500 patients were enrolled in that. And we created this oversight committee for any COVID-related research because everybody was developing protocols. Multiple protocols were duplicated, and we wanted to make sure that there wasn't multiple papers on the same subject coming out from our institution with different data. We also wanted to make sure that the process was streamlined and people weren't competing for the same samples to the detriment of patients. As you can imagine, only a certain amount of blood could be taken, and we didn't want people competing for that blood. So all protocols were reviewed by this oversight committee, and we determined which groups should be allowed go ahead or not, which became controversial on occasions. In addition, the IRB, if it was COVID, did rapid review, and we got studies approved in record quick times, which makes you realize that when everybody's aligned, you can get HR, legal, and IRB to do things in this phenomenally quick time, which was pretty amazing. We still have those processes in place, and we have now created a COVID clinical trials unit, which will continue still overseen by Judy Aberg, which will do the COVID clinical trials going forward. Our biggest issue with that at the moment is that fortunately we have so few patients. And so we have to determine which studies we prioritize. And then as we wound down, and I'll show you some of the numbers, how do we pivot back to normal practices when patients are afraid to come in? And then now where we're at the COVID, the Center of Excellence for Post-COVID Care, the registry and the biorepository. And for that, we have specifically targeted enrolling patients that were admitted, including those that were in the biorepository and registry for inpatient. There are 500 patients. We've made sure we target them so we get longitudinal information on those patients. And also patients that tested positive within our system that were never admitted. As I'm sure is the same for you, a lot of the patients we didn't admit were actually very sick and have long-term sequelae from COVID. And a lot of the patients that were tested positive and went home did fine. So part of this is understanding why did some people do well and why did some people not do well. So to do that, we need to cast a broad net for inclusion in the Centre of Excellence Registry and Biorepository. So that's some of the organization that occurred in response to COVID across our 7 main admitting hospitals. So this is actually the volume. This does not include one of our hospitals because this is EPIC data, and Mount Sinai Beth Israel is not on EPIC. So we peaked in the first week of March, I've lost track of time, and we peaked at 2,300 patients across 7 hospitals. This is where we're at now. So much better, 188 patients as of yesterday, most of which are still PUI. Oh, sorry. It's doing something funny on me. So to give you a look and pictures about what this looked like. This is where I work on the Upper East Side, and this is New York when it's nice and calm. What was interesting, and everybody commented on it, was people who lived in Manhattan noted the constant sirens at night, constant ambulances going at the peak of the COVID crisis. I even noted, I live in Westchester, which is a kind of sleepy suburb. I even noted that at night and during the day, there were a lot more sirens, a lot more ambulances. My town is beside New Rochelle, which was one of the big peak. It was actually the first case in New York, and really it was one of the major peaks in New York. So just this constant sound of sirens at night and during the day. This is what we looked like after we surged, and this is Samaritan's Purse set up a 10-tent field hospital and ICU. They were really very, very impressive. There was a lot of social comments about the fact that we worked with Samaritan's Purse, and that was because they are from a very connected with a very conservative right-wing church organization in New York, and they are very anti-LGBTQ, which is very opposite to the ethos in our institution. So people were very upset that we partnered with them. But when you looked at the peak that we were looking at and the capacity we had within our system, what we really needed the ability and the lengths of stay in the ICU. We had to look at some way to decant the inpatient census, either convalescent or ICU cases, so that we could admit acutely ill patients. At one point, Samaritan's Purse were going to set up a field hospital in St. John Divine Cathedral. That stopped due to differences in opinions between the people who oversaw the cathedral and Samaritan's Purse. So to give you an idea of what the ICU looked like, it's really pretty impressive. That's the ICU. The person in the white coat is our head of Critical Care Institute, Rupa Kohli-Seth. So this is really pretty impressive for a field ICU. We also expanded our capabilities by, Catherine, you will recognize our lobby or not recognize our lobby. This is the lobby of Mount Sinai, and we developed our own field hospital in our lobby. This is it. So we were ready to, again, put convalescent patients into these units. Also, the the packages we started receiving of PPE, we were very lucky. We were beginning to run low on PPE, and through connections with our board, the New York Jets, a football team, sent their private plane to China and picked up shipments of PPE. So we were very lucky. We never ran out of PPE. We had plenty of PPE, but it was through significant negotiations. Our board actually wound up being tremendous. They have raised a huge amount of philanthropy. They in fact raised 70 million for research, which the dean then used to help us get various protocols up and going. He funded things like the development of the antibody that Florian Kramer developed. He's funded the biorepository. He funded the immune monitoring core, the Luminex assay for immune monitoring on our inpatients. Also funded the data group that were working with us so that we could get up and going immediately and not wait for external funding to be able to do that. Our head of the board has also started something called Fit for the Front Line, where people around America are doing various forms of exercise, getting sponsored and raising money to help the COVID response in our institution. So it really was an all hands on deck in our institution. This is Cap Powell, our head of pulmonary critical care. You know, there was constant requests for interviews. Oh, I need to wrap it up. Okay, thank you, Catherine. So he, along with the head of the sleep program, developed a mechanism to ventilate patients using CPAP machines. They also developed a mechanism to dual ventilate patients using which I know multiple people have, but they put a valve on so you could regulate pressure for both patients. Also, Florian Kramer, whose research is on influenza vaccine, developed an antibody which is now being used for quantitative assessment of response to COVID. There was also this need to rapidly onboard ambulatory doctors who had not taken care of inpatient acute patients for a long time, and also create this new teams for the inpatient service. I will go through this as quick as possible. We actually wrote up what we did and was published in the New England Journal around creating new teams. So we divided up between ICU and outpatient and allocated different departments. So I was allowed to take doctors from very specific departments, and critical care were allowed to take from, anesthesiology and other groups. So we didn't compete for doctors. And then we created this new inpatient service. There was a hierarchy. We created these not based on whether you were an intern, resident, fellow, or an attending, but based on how recently you had been on a medicine service and what your skill set was. And so we put chief residents and senior residents in charge of teams. And we had, I like this down here, division chiefs, associate deans were the lowest tier. We also had orthopedic surgeons down here as acting as interns. We recreated the inpatient service. What we also did was we totally reorganized our inpatient service to create geography. There was data from China that if you did absolute geography, i.e. if you had 12 patients and you put them in rooms 1 to 12, that you then didn't change PPE, you changed gloves, and it decreased nosocomial transmission of COVID. It certainly, then we had these teams working together with the nursing, with the respiratory techs, and it decreased anxiety amongst the healthcare providers. And the teams got to know each other, patients knew the teams, and it worked very well. The flow worked really, really well. So I'm going to go through this next part super, super quick.
Robert, sorry, if I could just intervene there. We're just a little bit concerned with time. If you could wind up.
I'll stop here if you want. I can stop here.
Yeah, I think probably over to Catherine if that's okay.
Yeah, that's great. Thanks very much, Barbara, and that was really good, really interesting. Can I thank all the panelists? We have probably just a few minutes for discussion now because we've gone significantly over time. But can I thank the panelists especially for answering many of the questions that have arisen through the chat function. I see many of the questions have already been answered. But if I can kind of highlight a couple of questions which have come up very predominantly, and in fact, Barbara, you might want to answer one of these.
I can't share, sorry.
Which is about the the comorbidities associated with COVID-19. So we've all read a lot about sex and about men being more badly affected. So maybe Barbara or Paddy, could you tell us, is that Paddy actually, is that the case in Ireland? And if that's the case, is it that men get worse disease, or are there more men get infected? Is it that they develop worse disease? And is that age related? For example, do older women have the same risk?
So we definitely saw the international pattern here, where we saw much worse outcomes in older men, more hospital admissions in older men, and I don't think that it's I don't think it's a product of them getting more infections, because we had an outbreak within the nursing homes. The nursing home gender balance was relatively the same, so you had a lot of people within one environment getting infected, and we still saw the predominance of morbidity, mortality affecting older men. So it must be a host issue. I don't think it's really been that well teased out, and it's certainly something that will need to be the focus of research. Just very quickly, one of the big challenges of doing research within that older population, though, is the consent issue. So the vast majority, for example, of patients coming into hospital from nursing homes, we weren't able to get engaged in studies because of issues around capacity and consent. And that's something that we're looking into in terms of looking at derogations for consent. But it's a very, it's a very delicate area when you're dealing with very sick people that lack capacity to start doing research on them. But I think there's something more than just the stats that's driving this, Catherine. I think there's something scientific basis to it.
And we've also seen a lot around diabetes, and it seems that people with both types, so it wasn't obesity, it wasn't a simple obesity type 2 diabetes connection, it seems to be type 1 and type 2. So are those patients who have more of the chronic complications that you might associate, for example, with the coagulopathies and those sort of conditions that ended up in poor outcomes?
So I think the coagulopathy picture, there was a few questions about it, is it driving end-organ disease? I definitely think it's in the mix. And you're right in terms of the interplay between obesity, the immune dysfunction that goes with obesity, and the interplay between coagulation, endothelial function, and the immune system is very clear. The one thing about the one really unusual thing about the coagulation picture is that in the post-COVID syndromes, we're seeing a lot of dysfunctional coagulation in people that are young, healthy, normal BMI, and it's going on for weeks post what would be an initial probably mild infection. So there's something really unusual going on around the coagulation that I think is almost unique to SARS-CoV-2, and I don't think we really understand it.
And are these people that were hospitalized or people that had a relatively mild disease as well?
These are people that post-COVID clinic, a lot of them are healthcare workers or GP referrals. So these are people that never saw a hospital. But yet when we start looking at that, we're seeing a small proportion of them coming through with really markedly abnormal markers of coagulation weeks down the track. Really unusual. Okay,
can I ask maybe Kingston, do you think that the world has turned and we believe in experts and we believe in vaccines again? Is that going to be a big challenge? I mean, I saw that only 70% of people in the US would use a vaccine if it were available.
Yeah, I think in Europe it's a little different. I think in Ireland we had some ups and downs in terms of vaccine hesitancy. I think if everybody had a vaccine right now, I don't think there'd be much hesitancy about it. So I think the vaccine is going to be our savior, and I really think if people don't accept vaccination, it would be a travesty. So I think people will buy into the concept of vaccination for this if we get a successful one.
Barbara, do you think it's the same in coming from the US?
Can I make it, Catherine?
Sure, sorry, Ultan. Yeah, go ahead.
Anti-vaxxers. So
yeah, so my biggest concern with the accelerated mode by which the vaccines are being developed is that there won't be enough time, and there may not be enough time to properly look at the any adverse events or any toxicity or things like that that's going to happen with those vaccines. If it ends up that we start moving really, really quickly, and we end up with some people getting sick or getting toxicities or adverse events associated with it, it will play massively into the anti-vaxxer movement, and it will cause a huge problem then in the use of vaccines for all other conditions. And this is my big concern, is that we have to be extraordinarily careful to monitor the safety of these vaccines all the way through. If we fail to do that, I think we've failed massively.
I agree.
That's a really great comment. Thank you. Barbara, do you want to make any comment?
So, you know, in the US, there's a large community of anti-vaxxers just for like established, what we would regard as normal vaccines. So I could see that there might be issues over here.
Okay, another question that's come up from several people is around, we've seen a lot in the UK that BAME people being disproportionately affected with adverse outcomes that seem to be dissociated from socioeconomic issues. So I don't know if we've seen similar issues here. Paddy, have you seen anything like that? Or Sam, either of you?
I guess one of the things about looking at it within the Irish context is that a large proportion of the people that were diagnosed with COVID-19 in Ireland were healthcare workers. And within the healthcare worker population, there will be a big disproportion of people of BAME origin within that. So you probably will end up getting a biased view if you do that as a straightforward analysis. The UK had a much more broad epidemic, and certainly the signals came out there. And I think that it's within that context that you're probably going to get more accurate data in terms of what's driving it. I don't think that we'll get anything that's other than a biased view if we look at it within the Irish context, unfortunately.
Yeah.
It's a good thing because we had less infections, right?
In Beaumont, we did an analysis of this, looking at all the cases we diagnosed based on their home address, and we're able to map that to Irish census data on the average wealth of the small group in the area they live, which is reasonably accurate surrogate of wealth, and didn't find any socioeconomic gradient in those who compared to the general population, or in those who died compared to those who didn't die. So that wasn't race or skin color or where you were born, but it was socioeconomic, which surprised us. And that was about a sample size of about 400 or 500. We had a huge number of cases.
If I can make a comment about the UK situation as well. So the data from the UK, this is the ISRIC 4C study, is clearly indicating that there is a very strong socioeconomic impact on severity of disease. But there is one group, the Asian group, that seems to be definitively associated with enhanced disease or severe disease in this group compared to other groups. So it's not necessarily origin, it is, it's not necessarily the genetic background of the individuals, but socioeconomic impacts seem to have a huge influence.
Okay, great. Thank you. So can I ask, are we ready for the next pandemic? Have we learned enough to do with the next one? Sam, we'll ask you, I think.
Definitely not, no. We've no plan for any of these. The reason why Asian countries have done so much better is that they had a plan for SARS-1 and for coronaviruses and for flu. We've nothing like that ready, but clearly we need to learn from that.
Do you think we're going to be so relieved when it's over that we'll just want to forget about it, or will we actually plan?
No, this isn't going to be over. This is going to be a new normal. It's going to go on for 5, 10, 20 years. And I hope we plan for pandemic flu and all the other possible infectious diseases that could transform our lives and society and healthcare system.
And what about the day jobs in the hospitals that don't involve COVID-19?
We've got most of those back up and running. We've got our surgery back up and running safely. We're screening everyone 2 days before you come in, and we're getting them home quickly and have taken precautions to prevent in-hospital transmission. So I think that's rapidly scaling up over the last 4 to 6 weeks, at least in my hospital. I'm not sure other hospitals are catching up on that, but we certainly are.
And can I ask, given that we were told this morning by one of your colleagues in infectious diseases that sunny holidays were completely fine, do you think we need to do what I've read that they do in Vienna or in Austria, which is test people on the way in?
No, I completely disagree with Jack. We shouldn't be going travelling for this summer. It's the wild Atlantic way in West Cork is my advice. You're just inviting more COVID-19 back into Ireland if we all travel to higher risk places, whether it's in Europe or certainly in America. Many states in America are still now getting 40 or 50,000 cases per day. So I think it's really important that we don't travel. So I strongly disagree with Jack. And we did a WhatsApp of the members of the Irish Infectious Disease Society of Ireland just earlier before this call, and all of the members except Jack disagree with Jack.
Okay, well actually what I was asking you as a follow-up was the idea of testing people in airports on their way into the country.
The problem is that they could still be incubating it. They could still be at an early stage. So we do not know yet at this stage whether a swab in the nose, as Barbara told us happens in Vermont, is accurate. You could be negative today, but yet in 3 days' time it becomes apparent. There's clearly an incubation period before you start shedding. And if you're in that incubation period, you clearly won't be positive.
Okay, well, we've now gone almost 30 minutes over. So can I thank all the panelists very much. I'll be chatting with you later because I've lots of really specific questions, but it was really great. Thank you for being so generous with your time and with your expertise, with your collegiality, especially with answering the chats as the discussion was going on. So I'll just remind you of the next two conversations in this series, which are next Wednesday and the following Wednesday. I think the titles of them should be up on the screen.